Okay, so let's continue on with this now. So P53, we said, plays a really important role in making sure that when you have double-stranded breaks in your chromosomes, that different enzymes can detect that there's something defective with the DNA, and then it can trigger P53 to do things like halt the cell cycle, um, or go ahead and initiate different enzymes to try to um, repair the damage. And if the damage is replaced or is repaired, then P53 activity is turned off, and then the cell cycle can resume, right? But just as long as there's damage, P53 is activated and the cell cycle is halted. So we had learned about this phenomenon in transforming cells called BFB mutations, right? Or breakage fusion bridges, where as the telomeres wear down on the ends of chromosomes, they may actually begin to stick together. And if they do stick together, then when the cell undergoes mitosis, if it's not actually stopped from dividing, then you're going to have problems, right? So let's take a look at this here. So if you lose P53 in the cell due to mutations, for example, you're going to get progressive telomere erosion that goes shorter and shorter and shorter with every cell cycle because P53 is not available to stop the cell from dividing. And then you're going to end up with these sister chromatids that then fuse together. And when the cell divides into two new daughter cells, right, they're going to be torn apart kind of randomly. And as we learned last time, other chromosomes are undergoing the same phenomenon. So you end up with this brown chromosome sticking to the green one. And then that breakage, that fusion, forming bridges and then tearing apart again, goes on and on and on if P53 has somehow been deactivated. And so this period then, as the cell is going through this, we would call this a period of genetic instability. So transforming cancer cells or transforming cells that are heading towards cancer are very genetically unstable. And so that implies, again, that if we look on the left here, we have a cell that was normal and then started to transform. As it divides, we learned that most of the daughter cells that are going to result from these divisions are actually eliminated because the cells do the right thing, so to speak, and can detect that there's something horribly wrong with them, and so they undergo apoptosis. But if some daughter cells survive, right, they go on and keep dividing and dividing. And with each generation, there is the potential for the genome to get progressively more and more messed up through this genetic instability. Right? Again, most of the daughter cells will do the right thing and undergo apoptosis. And that's why it takes many, many, many generations to build up the combination of random mutations that eventually will lead to full-blown cancer. So an important thing to keep in mind, though, is this whole intermediate phase, this period of genetic instability, it can only go on for so long before the cancer cell itself is so messed up that it can't even survive anymore. Right? Because obviously some of these genes, the cancer cell still needs to have intact and working properly just to be a living cell. So that's why we discussed this idea before, that at some point, a critical feature of the transforming cell has to be a set of mutations that it stumbles upon to reawaken the TERT locus. Right? So remember, telomerase is what adds additional copies of the telomere repeats on the ends to extend and basically replenish or restore the telomeres. So at some point, if those transforming cells stumble upon that, you can see TERT is woken up again, and then that puts an end to the period of rapid genetic instability. It's almost like you've now stopped or frozen the genome in whatever really messed up state the cell managed to survive to after many, many generations. And importantly now, this is a big problem, right? Because now the cancer cell is somewhat stabilized, and it will now exhibit all the types of cancerous phenotypes in a relatively now stabilized genome. So this raises an interesting point, and that is that Mother Nature and the whole process of evolution brings with it a lot of baggage. One way to think about it is that the more complex an organism becomes through evolutionary time, the more potential problems must be balanced. So if you think about what we talked about before, relating the lengths of telomeres and also whether or not the TERT enzyme locus is active or not active to replenish telomeres, it's always kind of a trade-off. So you have telomeres that are shortening with every cell division, which put a distinct limit on how many times a fully differentiated cell can proliferate. So if it's building up mutations in our bodies, it's not a big concern because those differentiated cells stop dividing and they senesce or are just physically lost, right? But of course, a big part of that is the fact that in our fully differentiated cells, the telomerase is turned off. The telomerase is active in stem cells that need to constantly rejuvenate or replenish itself, right? But in normal differentiated cells in our body, the telomerase is turned off so that the shortening of the telomeres can be used as a clock. So if you think about this, though, this causes an interesting conflict. Because our cells are always having limited proliferation capability, that means as we age, as we are alive longer, the wear and tear on things like our stem cells really takes a toll. But at the same time, because we expect to live for several decades, it is important that we constantly have these shortening telomeres because they help make sure that differentiated cells, the 11 trillion or so differentiated cells, or 20 or so trillion differentiated cells in our bodies, right, don't actually have time to stick around and become cancer. So what do I mean that this is a trade-off between preventing cancer and accepting age-related damage? So a lot of that has to do with thinking about our life expectancy. This is actually a really amusing kind of article. I think it came from a Canadian popular science journal. But it basically talks about reflecting on the life expectancy of us as a species through our eons of evolution. So if you think about it, starting here about 30,000 years ago, which I think is a rough estimate of when Homo sapiens, us, we sort of separated from our closest ancestor at the time, the Neanderthals, right? So about 30,000 years ago, if you thought about Neanderthals, this is kind of funny. The average life expectancy would be about 30 years, right, for a Neanderthal, for a primitive human. And that's because most of the time, they would die of everyday things like injuries caused by rock falls, which I think is kind of funny that they mention that, or hunting accidents, or conflicts with each other, right? There's food malnutrition, or not enough food, so there's always a concern about where you're going to get food from. So as a result, the life expectancy was just 30 years. If you jump ahead to about 8500 BC, you see that life expectancy now goes up to about 38 years. So that's about as long as somebody who's born then could be expected to survive. And that a lot of that has to do with the invention of agriculture, irrigation, humans coming together to live in societies like villages and cities. If we move up to 500 BC, now we're getting into the classical Greek and Roman era, right? The life expectancy actually drops a little bit. Why? Because now we have really big cities, and that means different diseases, communicable diseases, can spread through populations much more rapidly and have a bigger devastating effect on the number of people who are actually around. So things like tuberculosis, typhoid fever, smallpox, right? Those would run rampant through there. Then through the medieval time, to late medieval time, you see the life expectancy goes up and down again due to things like the Black Death affecting it. And then if we zoom ahead to the mid 1800s to the 1900s, the life expectancy is now stabilized at around 40 years old. But again, because we don't have yet modern medicine, right? And so living in big cities, right, that puts a real strain and real threat on us by catching different types of diseases. But this is the interesting part up here. Once you jump past the 1900s, life expectancy shoots up. Why? Because we have better healthcare, sanitation, living conditions. We now have nutritious foods and things that are relatively easily accessible. And then if you look at today, our life expectancy, 82 for men, 85 for women, because in our modern society, right, we're not as worried anymore. We have modern medicine, we have lots of access to high quality food. And so now, interestingly, the big concerns that threaten our health are no longer things like typhus or smallpox. Instead, they are things like heart disease and cancer. So this is an interesting point here, that if you go back in time 30,000 years, which in the greater scheme of evolution, 30,000 years isn't that long at all. If you think about it, right, things like cancer and heart disease were not the primary concerns. Because again, human beings at that time did not live long enough to worry about cancer. And they did not have sort of some of the bad lifestyle choices and overly fatty foods that we have now to worry about things like heart disease. So in a way, our life expectancy now and the problems that come with it, like cancer and heart disease, are really a function of us just living longer than what Mother Nature actually designed us for. So let's consider the role that aging plays in cancer development. Our body is constantly making a choice between being okay with the damaging effects of aging because it means we are pushing away the threat of developing cancer. Fully differentiated cells, we've already discussed this, they stop dividing to prevent transformation, right? So that those differentiated cells that are actually getting hit with mutations, they stop dividing anyhow and are probably just lost from our bodies. But remember that there are relatively few stem cells that are around to constantly replenish those fully differentiated cells. So those cells have to stick around, right, to constantly resupply all the differentiated cells in our body. We looked at this series of photographs before, but this is your outer skin, your epidermis. And we said that if we look at these darkly stained purple cells, these are the keratinocytes, right? These are the outer dermal cells that make keratin, and then that keratin, after the cell dies, just contributes to this thick outer layer of protection that our outer skin keeps our outer skin integrity, nice, and a good physical barrier, right, against the outside world. In an 18-year-old, the keratinocytes are very abundant. By the time you reach a 76-year-old's body, right, far fewer keratinocytes are around to replenish the skin. And that's why the elderly, it's not surprising, right, their skin is much more fragile, prone to breakage. But why? Right? Because after all, it's the stem cells that are sticking around and constantly replenishing these. If you think about it, somewhere down here in the lower parts of these photographs, you would find, 1 in 100 or 1 in 500 of these cells are actually the stem cells that are constantly dividing and replenishing the keratinocytes that you see above in the photo. After, let's say, close to 8 decades of life, if you think about it, you've probably lost a lot of stem cells along the way because of different things, like those stem cells just die for various reasons. While those stem cells are dividing, maybe they make big mistakes in cell division. And so the 2 daughter cells that result both undergo apoptosis. They do the right thing, but then that means you've lost that stem cell. And just think about the stem cells that haven't died, that are sticking around. every decade that goes by, how many additional random mutations those stem cells are now carrying with them as baggage. And that means over many, many decades of life, your stem cells simply aren't the high quality stem cells that you had when you were a teenager. And so their proliferative ability has gone down, right? And think about it, all of the keratinocytes that come from one single stem cell, by definition, will carry the baggage of all the mutations that that stem cell has built up over time. Any questions about that? Okay, so let me just stop this.